And let me share PowerPoint. Okay. So can you see my slides now? Is just one of you confirm? Yes. Okay. Awesome. So okay. So what's the main objectives of today's lecture? So as you can see today, we are at the level where we already got into the DNA amplification steps. But now we want to do really a practical example of how we can obtain a gene from the GenomeBrowsers, the NCBI or the Ensembl. We would like to identify the sequence. We look at the gene, where's the exons, where's the introns, if it has any SNPs or single nucleotide polymorphism, for those who forgot. And then we will go back after we retrieve the gene, and we will make a scenario that we would like to amplify a part of that gene by PCR. And in order to be able to do that, we will make a primer design. So we will go through the primer design together. As you remember, we discussed all the details of what is the PCR, how we can use it, where we can use it, what are the details, what is the steps of the reactions in terms of denaturation, then the amplification step, and then all the details we have to take in considering a PCR reaction. So we are not going to repeat this today. We mentioned what are the main parameters we have to take care of and how we perform a reaction. And we discussed the 3-4 different steps in the PCR design. And if you didn't watch the videos, I would like to remind you that these videos are really, really useful for you. I strongly recommend that you guys go and watch these videos, because every 2-3 slides, I'm adding a video for you so that you can imagine what's happening. But when you see it in the video, it looks much, much easier, because this is a practical course and describing the steps doesn't really help much. So let me just refresh your memory about how genes get translated into function. So you all know that the genes, in principle, residing in the DNA sequences on both strands of the DNA would be transcribed into messenger RNAs, and those messenger RNAs are translated into proteins. Now, this gives the cell multiple level of control. The first control would be how much we are transcribing DNA into RNA, and then how much and how we are translating the messenger RNA into a protein. And therefore, you have a control on all of these steps, which in principle, at the end of the translation process, we end up having the functional units, which are the proteins which perform the function. So we mentioned this last time. Now, if we dive closer into the gene sequence, the genes consist of, in terms of the coding or the open reading frame, the open reading frame normally contains exons that are flanked by introns. So the exons are the coding part of the gene, and the introns are the non-coding part of the gene. Now, when the DNA is being transcribed, we cannot take into consideration removing those non-coding genes. So the messenger RNA, in principle, is transcribed as both exons and introns. But then we end up having the RNA splicing to get the mature RNA, which will code eventually for a protein. And this all happens in the nucleus, and then it's exported out into the cytoplasm where the translation takes place. Now, last year, I had questions about where the translation takes place, where's the transcription take place, and where's the RNA splicing take place. So be careful that this could be a question for you. Now, looking at the promoter itself, at the gene, in order to be functional, it has to have a promoter, which is the initiation of the transcription. It also requires some flanking regions that are not translated to the protein, and these we call them untranslated region on the 5' end and the untranslated region at the 3' end. Then the exons, the introns, of course, the start and the stop codon, and some sort of polyadenylation that happens at the 3' end to protect the messenger RNA, and some sort of enhancement for at the 5' end as well to protect the RNAs and increase the stability. And finally, there are some other factors that are not presented here. These are sequences that are probably away from the gene, the enhancers and the silencers. Okay, now for the process of gene transcription, once we have the RNA transcribed, it could be spliced in different ways. So here are like two scenarios for you in terms of the RNA splicing. So we have here exon 1, orange, exon 2 in blue, exon 3 in green. One way we can splice this RNA is by getting rid of this guy. So we skip the blue exon. And by skipping the exon, we have now a messenger RNA that has only exon 2, exon 1, and exon 3. You can imagine that we had another version of the messenger RNA that have exon 1, exon 2, and exon 3. So you already have two isoforms of the same protein. Well, exon splicing is very complicated, and it could take many, many different ways. This is another scenario here where you only take a small part, which is a truncation of the exon number 2. So you have exon 1, a fragment of exon 2, and another, and exon 3. So this is going to be different protein compared to this guy here. So it's good to keep into consideration that RNA splicing is giving variability in terms of the outcome of protein translation. So this is a schema that represents what I explained here. So you have the untranslated regions, which is supposed to be here, upstream of the gene or downstream. You have the start codon where the transcription initiation starts and the transcription termination, where the stop codon is. And not necessarily that. So we have the RNA coding part, which is mostly the exons. And per se, I would like you to remember that the upstream of the gene is toward the left. So anything toward the 5' end. And the downstream is normally what we call downstream toward the 3' end. Now, since we have two strands of DNA, we call one of them a template and one of them non-template, or one of them positive and the other negative. So you have to be careful when you're looking at your gene in a genome browser, and I'm going to show you that, to be careful where is it being transcribed. Is it on the positive strand or on the negative strand? So one example, so for the 3' and the 5', they are not really very well annotated in this figure. I just put for you the markers here. And by convention, most of the time, the expression is being followed by 5' to 3' end. And the regions, as I said, to the left is the upstream and to the right is the downstream. Now, this is an example for you where the positive strand is from the 5' toward the 3' end and the upper band and the upper strand, and the negative one is the again, it's always from the 5' to 3' end, but now it's on the left end. This is an example where transcription is taking place on the lower strand. So you can see the transcription started here. This is the first ATG, and when it's translated, it's going to give us a protein that is being transcribed toward the left. So this is not something very uncommon. We have many genes that we know that are transcribed on the negative strand of the DNA. The important thing is that you know it's always from 5' to 3'. Don't mix things up. Now, because today I would like to do some practice, I don't want to give you too much theory. And I will be going too many times to different websites. So I want you to follow me, please, carefully, because following me on these, it will make it very easy for you to solve the assignment questions. And it will not take you more than half an hour after following the details on this lecture. So one of the most known and the biggest database that we have for the biotechnology genome related information is the NCBI, which is the National Center of Biotechnology and Information. So it has tons of information about the genetic material, the databases, the proteomic information. There are multiple categories in this website, like the gene bank, the PubMed. The gene bank is mostly every single gene that has been cloned, sequenced, verified. You can find it there. You can find all the information, how it was done. In the PubMed, you have also what is one of the tools that can link you to all the publications that has been done on a certain gene. The reference sequences, it will link different genome databases, and which it will show you the genome, the sequence, the transcripts, the variations on those transcripts, and the proteins. And there are many other tools. Like now, for you particularly, I'm not going to make it so complicated, because I don't assume you that tomorrow you're going to go somewhere and start performing an experiment where you need to dig yourself in the NCBI without any support. Plus, some of the things that I'm going to show you probably would be covered better in a bioinformatics course. So I'm just giving you the basics here. Now, something you have to take into consideration that in the NCBI, there are accession numbers to each of those genes. So in other words, there's a unique identifier for each of those genes or sequences that it doesn't overlap with any other gene. So if you write an accession number in NCBI, it will bring you out only one product. Now, the D number could be in different versions because there are different information that has been introduced, but the original number, it stays exactly the same. So, and that's what it said here. Now, one thing you have to keep in mind and directly understand from the name itself, like if you see accession numbers such as NM and then these numbers, this directly tells you that this is the messenger RNA, standing for M for the messenger RNA. And the same here in case you have the MP, so that will tell you directly this is a protein. So if you're looking on the website, you will directly know that you're talking about a messenger RNA and therefore the coding sequence, or the protein and therefore the sequence of the amino acids. Now, the second browser that we will use, and I find this a little bit more friendly for what we want to do, is the genome browser, the Ensembl. It has more or less similar database. So there's a lot of genomics that has been integrated. So there's the human genome, the mouse genome, like more or less you can see the same things in NCBI and Ensembl. Maybe Ensembl is a little bit one step ahead by creating a more friendly profile on the web. So you can click and find directly the exons, click and find directly the introns. With one click, you can get the SNPs. But again, those two browsers can get you almost the same information. For example, if you look for the BLAST, and BLAST is the function, if you have a sequence of 10 or 15 nucleotides and you want to know where those 15, 20 nucleotides come from in human genome, you can use the BLAST. It will tell you that it belongs to this gene. It can be found in this gene. And there's a possibility that there's a similarity in the sequence to these 3 or 4 genes. So both browsers can give you this functionality. Now, for today's application, I want to quickly go with you on the Ensembl, and in few slides, I will go directly to the website and I will start doing the retrieving of the sequence with you. But now these are only slides, so there are no clicking. So if you look, you go to ensembl.com and it's gonna .org, and it's gonna directly go to this website. Obviously, you can choose more than one genome. So I am now in the human genome. There are multiple versions of the human genome. Every 3 months, they update one. So you might have something having 38.P14 or 38.15. So this means the human genome has been updated. It gives you a summary for the gene we will discuss today. Is the periostin, which is a gene that plays a role in fibroblast and bone formation. And it directly tells you from the name, just to remind you that whenever the gene is written in capital letter, all of the letters are capital, this means it's a human gene. And then if you write it with capital P and then the rest small, then it would be a mouse gene. Then there's a lot of information already on this first page. It tells you the unique ID for this gene in the Ensembl. So it stays ENS G, so the Ensembl genome, and it gives you this unique number. And it gives you direction to the accession toward the NCBI. We will click on this later on, and we'll see what will happen. Then it's telling you the gene names, and if there are more than one name, it's going to list all the names for you. It's periostin, PN, OSF2. It will tell you the location on the gene, on the genome, on which chromosome. So it's on chromosome 13, and it gives you exact location between which base pair to which base pair. It's telling you that it's on the reverse strand. So we know that it's going from from right to left in terms of the transcription. It tells us that there are 10 transcripts, and if you count these are the 10 transcripts. There are multiple splice variants. It happens to be common with human and other orthologs, like in primates, in fish, in mice, and in many other animals. Now, if we look down, we will see that automatically we got the most common being shifted on the top. Because if you look, most of the parameters are known about this. We know that it has a 3,301 base pairs. It codes for a protein that is 836 amino acids. So the protein coding sequence is there. If you click on it, it will directly take you to the protein coding. And there are matches between these proteins. So there's another database for proteins. It's called UniProt, if you haven't used this before, in the Swiss Bioinformatics Institute. We are not going to go to these details. Today, we are just discussing the coding sequences. And then we have the ref sequence that you can find in NCBI, where you can get the messenger RNA. So you can click to this, and it will take you directly to NCBI. Now, in terms of the biotype, you can see that some of these transcripts code for protein, so it will tell you this is a protein coding gene variant, and some of them they don't. So it says protein coding is not defined. So probably there is no coding; it's only a messenger RNA or non-coding RNA. Okay. Now, if we click on the NCBI connection, so recall that there's an ID here, that 16953. If you click on this, it will take you to a page like this, where again, it's gonna give you most of the information. It's periostin, its location, it's that location 16953, the name, the different names that are applied in which chromosome. It's on the Q on the chromosome number 13. And then it tells you more information here, that if you want to get more information about this gene, when it says curated, this means we performed tons of research. We have cloned the gene, we know the sequence, and we identified all of that. If it's non-curated, probably a software or algorithm that has predicted the gene and we didn't confirm it yet. So be careful that most of the time, if you want to do something, you have to look, it's curated. Now, if you click on this one, it will take you to the NCBI. More information about nucleotide, you can find here. All these links are clickable, and it adds you to the next page, and you can get the sequence. So if I click on that, it will take me here. Be careful all the time to double check that you are really having the ID of the gene, the primary source of the gene there. Otherwise, sometimes there will be multiple versions, and then you will have a problem in your experiment. So if you go back and check the gene ID, let me just remember, where was the gene ID here? I can't see the gene ID here. Oh, gene ID 10631. The gene ID 10631. So that's the same ID. Anyway, if you click on those links, there's no way that you will miss. But if you're looking on the NCBI, you have to be careful that you got there, because there might be multiple versions there too. And again, here, it will tell you a lot of information about the gene and how you can retrieve the sequences. Now, I'm not going to go to NCBI today, probably. I just want to make sure you got very well grasp on the Ensembl, because that's what you need for your assignment. If we need to do more on the NCBI next week, we can. So if we click on the NCBI, sorry, let me go one. If we click on the NCBI primary source here, it will open a format like this that will have all the information you want within the NCBI. We will see this together. Now let's go back to Ensembl. So if we want to check one of the transcripts, if I click on the first transcript, which is this guy here, and I will go back a couple of slides so that you can see. So it's this guy, the 47.9. So if I click on it on Ensembl, it's going to give me a summary of that gene, and it give me a visual summary as well. The visual summary is showing you first that the gene is around 36.18 kb, 36.18 thousand bases, or 36.18 kilobases. And it's on the reverse strand. And you can tell that there's a lot of these dotted line, and there's the connection. The connections are normally the introns, and those are the exons. So it tells you statistics. Exons, there are 23. So if you count these lines, there will be 23. The length of the transcript is 3,301 base pairs. So out of this, we will have only 3,000 coding. And then the translation of these 3,000 will give us a protein of 836. Exactly the same information that you found there but more detailed. Okay. And again, you have the links to NCBI, messenger RNA, NCBI, protein. Remember, NM messenger and P is protein. And the protein identifier in UniProt, which we are not dealing with. Now, if you want to get the coding sequence, you click on this guy. Okay. Another thing is that when we click on this transcript, another page open here, and it tells you this is the periostin 203. So that's periostin 203. And now you can see the exons, the cDNA, and the protein of that. So if we click, we go down and we find all the information we want about the gene. So it tells us the upstream sequence, the 5' upstream sequence that is untranslated. And then it tells us where the first exons start and where it ends. And then where first introns start and where it ends. So all this information is very well documented here. There's a lot of SNPs, there is a lot of stops, because there's a lot of variability. I intentionally chose to show you this this gene because it shows you too many parameters. Like there's splice donors, splice variants, there's a missense mutations. All that, if it exists, you can easily tack it down here. Now, if you are on a gene that is being transcribed, obviously in the first exon, you have to be careful that it's the one that is going to give you the start codon. Where is the start codon? Where, like I located 2 here. One of them would be that or that. And again, the variability in terms of the proteins of the variants, it could be because the start codon is different. So it could be this one or it could be that one. Now, not to waste too much time, I want to revisit quickly the primer design. And why I'm teaching you all that to go to the Ensembl to get a coding sequence, because imagine yourself you want to clone a gene. And I'm very confident by the end of this lecture, you will be all of you capable to go download a sequence of a gene and clone it somewhere, at least in silico, by applying a primer design and cloning into a vector. So the primers, we need it in order to initiate the process of amplification, because the polymerases, they cannot simply fly into the DNA and bind somewhere and start duplicating or amplifying the DNA. They need those small sequences where the 5', 3' are linked to are annealed to a DNA, and there is a 3' that is free, where the polymerase comes and binds and quickly start the process. And then the primers are so important because they are the sites where we specify where we start the amplification, where we end the amplification. And their design is very important because if the primers are designed well, the efficiency of amplification will be great. And if the primer design is bad, then the amplification is not going to be great. And remember, in the previous lecture, I described what are the parameters of a good primer, a good pair of primers. And we have to avoid those secondary structures. So we have to think about a primer about 18 to 24 bases, GC content 40 to 60%. Try all the time to have the first couple as a GC because they have 3 hydrogen bond in the primer, because that will make the binding between the DNA and the primer much better. The melting temperature between 50 to 60, that's because most of the annealing take place at this range, and the polymerase normally work at a range that is a little bit higher. So you want your primer to attach well by the conditions where the polymerase is performing the amplification. You don't want to have more than 5 degrees difference between the 2 primers, because each time you need to design 2 primers, right? One from the right, one from the left, and avoid these secondary structures. Now, we would imagine that we want to amplify this piece of DNA. And in the assignment, I'm going to ask you, and I'm going to do it for you right now, of course, on a different gene, but I'm going to ask you, go and retrieve a gene of interest. I give you the ID of that gene. Tell me what is the accession number? How many splice variant are there? How many amino acids you can get out of that gene after transcription? Show me how many introns, how many exons, and amplify, for example, the first 3 exons. So in this example here, what you see is that exon 1 is in black, exon 2 is in blue, and exon 3 is in black. So if I want to amplify the 3 first exons, I need to design a primer that sits here for forward, and another primer that sits somewhere between this one up to the CAG on the lower strand. And then I would need to add some restriction sites to this primer. So for example, I added here a BAMH1 so that after I finish, so on the first primer and on the second primer, I added different restriction enzymes. So I added EcoR1 on the second primer and BAMH1 on the first primer. The reason is, after the PCR is done, I can digest the PCR and it can give me a cohesive end that I can use to clone. And for the restriction enzymes to work well, they need the feeder. And I put the description for you here. The feeder is because most restriction enzymes need to sit on 4 to 6 nucleotide overhang for efficient cutting. So if I just put the restriction site only, the enzyme is not going to cut well. But if I put something where the restriction enzyme can sit, and then it can bind very efficiently to this site and cut through the site. So this is just like a handle for the restriction enzyme. We add it most of the time, like GAAA. You can see it's the same here. Now, for the forward primer, I don't have a choice. I have to start with the 80G. So I took the first 20. And then for the reverse, again, I don't have a choice because I want the CAG to be amplified. Now, for the reverse primer, there is something very important you have to keep in mind because you need the primer to be reversed and complement. So we are going to take this part here, the AA, G all the way till the end. And then first we will make reverse and complement so that we are working on the negative strand. And then we will add the restriction enzyme and we will add the feeder. And then you have the enzymes, the 2 primers are done. After that, what we will have? We will have a PCR product that looks like this, where we have a BAMH1 site on this and an EcoR1 site there. So if we digest this, if we take this PCR product and we put 2 enzymes at the same time, the BAMH1 and the EcoR1, so it will give me a sticky end. And if I take a plasmid like this one here, I cut it with BAMH1 and EcoR1. Again, BAMH1 is on the 5' end and EcoR1 is on the down 3' end. Then I will end up having my gene inserted into a plasmid. And I will already have a clone out of this. Okay, I'll stop here. I'll take few questions. And then I will go to the website to show you how to retrieve the sequence and how to perform the primer design on the website as well. So I'll stop sharing now. And I'll take any questions. Questions?
in relation to the assignment, is it already posted or might be posted?
No, assignment is
Okay.
not posted yet. It's
Okay.
going to be posted after this lecture. Yeah.
Okay, cool. Thank you. Everything was pretty straightforward. So
Okay. So let's go to the website, the Ensembl. So together. So just give me a second to share the gene. Okay. Okay. Share the genome browser. So if you go to the website that is in the link in the slide, useastensemble.org, and then you will have this page. Depends on the gene that you are working on. For today, we are going to discuss about the periostin gene in humans. So you would like to choose the genome that you are working on. Sometimes you are doing comparative analysis. Ensembl can allow you to make comparative analysis between different genomes like mouse, human, zebrafish, pig, cow, dogs, so all that. So for today, we'll click on a human, and then you get into this page. And again, it tells you that now we are on the genome assembly GRC H number 38, and this is the version you are using. I'm saying this because every 3 months this is change, and you might want to go back on the website and look for the version that you were working at. You can go back into the version 37 and 36. They are all available on the website. So if I want to use the word periostin, if I write periostin, I can retrieve the gene, and directly it tells you here periostin human gene. Now, if I go back and if I know the gene PBOSTN and I say go, it will find the same thing, periostin gene. Okay, so either the gene name or the contraction. So if you click on this one, you get to the page that I showed you in my slides. Again, it's showing you here that now this is giving you a whole summary of the periostin gene. This is the ID for the periostin in the genome browser, in the Ensembl genome browser, the description, and the link to the NCBI, the name, on which side of the chromosome, and you can read here, it tells you exactly where on the chromosome. So if you click on this one, it's going to be super complicated, but it eventually takes you exactly where the DNA on the chromosome number 13. So it shows you the site where the chromosome is, and you can play with this. I doubt you will need this at this point. So let me go back, but I just wanted to show you that you can go very complicated on this. If we go back, it tells you it's on the chromosome 13 location, and it tells you that it's on the reverse strand. Transcripts, the transcripts are there. So for today, I will click on periostin most known, and most of the time, the Ensembl is gonna kind of list those genes based on their significance of how much we know about them and which one is being mostly studied. And this is the most studied, and we have lots of information about it. So if I click on it, you can see already I have another button, another panel that appeared here, where I can see most of the information. Nothing really changed. But then the page changed from periostin, which is describing the gene in general, into the transcript, where you can get the exons, the cDNA, and the protein information. And those are very important for you to do, at least till now. So the transcript has 23 exons. If we scroll down, you will get to the image that I described for you before. So you will get the image for the gene. So if you're working on a gene the first time, you can export this. It can be represented. It tells you that it's on the reverse all the time. It's telling you this information again and again. So there's no way you can make a mistake with the information presented. Now, if I want to get into NCBI, simply by clicking, it will take me directly to periostin. So Homo sapiens, this is for human, periostin gene. And again, all in capital letters. So this is the periostin for human. And it tells you that this is transcript variant 1, because we chose transcript variant 1 on the list on the Ensembl. And that's the reference sequence. Now, there are multiple ways you can get the information in the FASTA format or in other formats. Again, if you look at the information here, you will see the locus. It tells you the exact number that is shared between both Ensembl and NCBI. And you can see that here, NM006475.3, that's the accession. So whenever we had identified that gene and gave it an accession, which is the unique identifier, it was 006475, which is there. Now, every time we add more information, more research, more data, we end up changing the versions. So now we're at version 3, but the ID and the accession is this one. So if I ask you in the exam, is the accession number this or that? Be careful that the accession number is the one that doesn't have a dot. Okay. Now, if you scroll down, again, we have the same information here, 3,301 base pairs of messenger RNA. And if you scroll down, you will get tons of information about the papers that has been published about this. And keep scrolling down, you get into the gene information, the start to end, the gene ID, the unique identifier that has been given in terms of nomenclature for the gene. And you can see on this side, you can get the exons and the introns. And then you can get the coding sequence. Myself, I don't do that. I prefer working with the Ensembl. So I just want to go back one step. So I close this guy and I will go back to this guy here. So I just showed you the route you can go to NCBI and get the information. But for me, I would click on exons. So if I click there, it opens this guy here, and then I would click on exons. And now you can get down to the page where I showed you. It tells you this is the untranslated 5' end upstream, and then you can get all the exons and the introns. Okay, now imagine it's very like you can see, for example, at the end of the transcript, there's a lot of 3' untranslated region. You see, so that's the intron number 23, and then there is a lot of the untranslated regions. Now, if I want to get into the coding sequence, which is the cDNA, I would click on that one. And again, just scroll a little bit down, and it's going to give you the coding sequence for that DNA. It tells you the first ATG is there, you see, and you can download the coding sequence from here. Okay, now, obviously, if you are doing analysis of genes like this, and I intentionally, again, chose something complicated so that you can see more than one thing. If you want to get the protein sequence, you can get it from here, click there, and directly you can go down and get the protein sequence. And the protein sequence will be identified exon by exon. So exon 1, exon 2, exon 3, exon 4, exon 5, exon 6. They just use two numbers that that alternate between the exon from one exon to another. Okay, so you can download the sequence. You can blast the sequence to check if it's common with other genes and stuff like that. Now, the other thing you can do, if you are on the transcript, you can click on the coding sequence here. So if you click on the coding sequence here, it takes you out into the NCBI. And scrolling down, you will find similar composition of the 23 exons, but now you have that in two colors again. Exon 1 in black, exon 2 in blue, exon 3 in black, and all the way till the end. And then it gives you directly in the same page the translation of those messenger RNA into RNA, into proteins, sorry. Okay, so imagine we want to work on this and we want to amplify these three genes, right? Okay, so I'm going to copy these three genes and I'm going to go to design a primer. So in order to design a primer, first I need to identify, well, I don't have too much choices because I will take a chunk here around 30 base pairs, and I would go to a website. I do go to this website, which is called Oligo Calculation. It will make a calculation for you in terms of the parameters that we discussed about the primer design. If I paste it here and I calculate quickly, it tells me that I have a 30 base pair. I don't need 30. I just need 20 to 25 maximum. So I can shop already 6 to 9 base pairs. So I shop that and I calculate again. Now I have a 21 base pairs. Now the GC content is 38%. We should be at 40, but let me go back one step and I show you. So I'll shop 1, 2, 3, 4, 5, 6, 7. I will calculate. You see, I have 23 base pairs and the GC content is 35. Since this is the ATG, I can't touch this part. I can do a removal of another T and I calculate again, it's 36. I will remove that other T and I will calculate again. And now it's 38. Now you can, if you want to make it perfect, you can do this. You can add a G or a C and then calculate again. And now it's a 41%. I don't do this normally, but it's possible to be done. Okay, so let's say we took this G and add it, but be aware that in my slides, I didn't do that, so don't get confused. I calculated, the length is 22, the GC content is 41, the salt adjusted, because your buffers, when you do a PCR, you have lots of salts there. So the salt adjusted melting temperature is 58, which is between 50 and 60. That's amazing. And then self-complementarity, you don't have hairpins, you don't have any complementarity between the primer itself, and it's not gonna anneal to itself. So now I will copy this part. And if you remember in my slides, when you go back to my slides later on, you will see that this is the sequence that I chose for a primer, but without the G because I didn't add the G for adding the GC content. Okay, so primer 1 is done. Let's go back to the transcript again. And if you remember, we want to amplify everything between exon 1, 2, and exon 3. So I'm going to take this part here and recall that the CAG, I need it. So I can't chop anything from this side. If I want to play, I want to play from this side here. Okay, so I'm going to copy this part till the G, copy. Now, I'll go again to that website. Oh, go back. and I will paste it. And I will do a calculation. GC content is 56. It's between 50 and 60. It's still okay. But what I can do is, for example, remove this G and calculate again, and it's 54 now. If I move the two A's, that doesn't help. If I remove the A and calculate, it will be higher. So it's better I keep the A. And now I have a 24 base pairs, 54% of GC, and the salt adjusted. Now this is a problem because this is a bit high. Okay. And when you get to such a situation, probably you can't do much because you have to play with the PCR parameters, make the reactions a little bit different on the bench, because you don't have a choice except using these two types of primers. Now, first thing you want to do for the reverse primer is to swap the strand. If you swap that strand, it will flip-flop to reverse complement. It will give you directly the reverse complement. So that is the one that you will need to copy-paste. And that is your 5' end, and that is the 3' end of the reverse primer. And then all you need to do, exactly as I showed you on the slide, is to add the restriction sites and the feeder. Okay. Okay. Now, just let me give you a scenario where it looks like the primer is not good. So let me take this. Copy. And if I calculate, you will see GC content is still full, but it's very high. You can already tell that this cannot be a primer. Bear in mind that sometimes you have to design primers like this. I did before a PCR where the primer was 100 base pairs. So this works. Now, one thing that when you check the self-complementarity, like here, it's amazing. There is still no self-complementarity between the primers. I want to try again to find one that gives you self-complementarity. So if I go back, apply, calculate, self-complementarity. Yes. So now it can tell you that there is a potential hairpin formation, meaning within the primer itself, you can see that you have the CCTT, and then this A here is gonna bind to anneal to the T, and then that G is gonna anneal to the C, and then that T is gonna anneal to the A, and the T on this side to the A, and the C to the G, and therefore the primer, it will bind to itself. So this is a hairpin within the primer. So most of the molecules will be doing this, and your PCR is not gonna work well. The other problem that you can see is the self-annealing, meaning there's a fragment of that primer, which is here, is gonna anneal, sorry, this part of the primer. It's the same sequence, exact same sequence, but now this molecule of the primer is gonna bind to that molecule of the primer because of the annealing of those red base pairs. And therefore, when you have this, you have to avoid using the primer if you have a chance. Okay. Be careful just for swapping the strand when you do the reverse, and then it's straightforward. What else do we need to do? We need to know what are the restriction enzymes in this fragment of DNA. So in order to know that, we go to another website, and all these websites I already gave to you on the slide. If we look on the NEP cutter, you just paste this and you say find me all the restriction sites on this gene or fragment of DNA. Why this is important? Because I can't use any of the restriction enzymes for my future cloning that exist in here. Because if I use those restriction enzymes, let's say I use FAT1, then it will cut within my gene. Or if I use BCC1, it's gonna cut within my gene. So I must have a restriction enzyme that doesn't have a cutting site within the gene of interest or within the fragment I'm amplifying. And in order to get that, you simply go to the enzyme list. And here, it tells you which enzyme has 0 sites. Like, for example, the ones that we used are the BAMH1. BAMH1 has 0 restriction enzyme, so I can use it. And it tells you what you have to add. So if you go back to my slide, you will see that I added GGATTTCC on the 5' end as a BAMH1 restriction site. If we go a little further down, EcoR1, also there is no sites, and you can take this, copy paste it from here, put it to your primer, and then add the feeder, and then you're ready. Now you are able to design primers for any PCR product you would like to do. Okay, I want to stop here again to get any questions. No questions. Everything is awesome. Okay, so we can carry on then. Okay. Okay, so then let me go back to my PowerPoint and quickly describe. Stop sharing that, and then I want to share the PowerPoint. Okay, so we went through the steps. We took the forward primer. We calculated, we were sure that there were no hairpins, there were no self-complementarity, there's no annealing on itself. We know the GC content, we know the melting temperature. For example, in the assignment, if I ask you to do a primer and you see that the salt, the melting temperature is high in the salt adjusted, you can define yourself, you say there's no chance that we can change anything in the sequence because we want to amplify this gene. But you can think about it in a different way, and I will give you a few tricks to help you think more about designing primers, especially for the reverse one, because in the reverse, you still have room. So probably you can design your primer to take part of this exon, although you don't need it, that's fine, but then you can have it there and have the restriction enzyme after. So probably you can take some of these T's if it can help you. We will discuss this in a minute. But this is how standard primer design take place. Now you can see this is the forward. We added the 5' to 3' and the BAMH1. We added the 5' to 3', the feeder. So you end up with a sequence from the 5' to 3' of the forward primer, and the same thing here. We have done first the sequence, we reverse and complement it, and then we added the EcoR1 and the other feeder. Okay, now after that, you have to be careful. You keep directionality, meaning if I have BAMH1 upstream or on the 5' end here, it has to be on the 5' end of the plasmid where I will clone. And the AQR1 has to be downstream, so that when I cut between these two and this opens, the BAMH1 will be here, this will bind to it, and the AQR1 will be here, and this will bind to it, and we end up having AQR1, BAMH1 with the gene of interest. Okay, probably the representation is wrong here. The AQR1 should be there, not there. Okay. Now, for the assignment, it's gonna be a very simple question. I would like you to retrieve a gene sequence. I will give you which gene is that. I want to know where is this gene. I want to know if it's on the positive or negative strand. I want to know the accession. Then how many exons, how many introns, how many splice variants. And I would like you to design a primer. For example, I want to say design a couple of primers to amplify exons 1 to 5 or 1 to 7 or 3 to 5, whatever. So you would do the amplification. And then in order to clone, you have to know which restriction enzymes you need to use. And these are all the websites that I used that you can use. And after you're done, you can validate your results by using the Primer 3+. The Primer 3+ can design this for you, but you will not learn to do it. You will learn nothing from Primer 3+. It's going to tell you this is a sequence and this is the other sequence. But then you will never know how it choose the information. Now, for the PrimerDesign, there's a YouTube video I added for you here. Please go ahead and watch it. Okay. Questions? To confirm, we can use any restriction enzymes that do not have existing cut. Absolutely. But, Gary, you have to be careful that it's better for you to use cohesive end restriction enzyme because it will make it so easy for you for cloning. Would we have to use these websites on an exam? I doubt it. You don't have time to do that. And that's why the assignment is meant to cover this part of the sessions. And for the assignment, when will we get, I'm waiting my TAs. I'll see when they will be ready. You guys are 150 students, so please be patient a bit. Any more questions? I still want to go a little bit on the NCBI since we have a few minutes left. Okay. So let me go back to the NCBI website then. Okay, so imagine you are choosing to find the sequences not on the Ensembl. You want to go directly to the NCBI. You can do that. You can go into the website and choose the gene, and I will go quickly in a couple of minutes. You have to choose the right genome, and you search for Homo sapien, let's say periostin. In NCBI, there's a very good characterization. So if the gene is very well studied, there is this window that shows up where the ref sequence is emphasized. So it will tell you the gene, and then it will tell you the reference sequence for this gene. It gives you the name, again, the ID of the gene that is shared between NCBI and other genome browsers like Ensembl. It gives you here links to the PubMed, where you can, in principle, why I can't see that. So here you can see the PubMed, all the papers that has been published about this gene. And under the ref sequences, which is, if you remember, the 006475.3, which is the most updated. It tells you the number of nucleotides, which is again 33,301, and the protein coding ref sequence, and how many amino acids, 836. It has only one isoform for this transcript, and it say the main select. You can see in green that others, they are really validated. It says that they are curated. This means we cloned them, we sequenced them, we performed experiments on those. But the main one that is strongly recommended to use is the main select. Okay, now if you go to the next, if you click on this, it will take you to the similar page that you came from the Ensembl. You can see that this is transcript variant 1 messenger RNA. Again, NM006475.3. Be careful that it's not another one. Similar information, you get the accession number and all the information that I've showed you before. And if you click on the coding sequence, and I'm going to go quickly to show you how you obtain this one, it will give you the coding sequence and the details will open down there. So let me quickly go to NCBI right now. So I'll escape here and I'll stop sharing and I'll go to NCBI transcript. Okay. So let me make sure that I'm sharing the right screen with you. Share. Okay. So if you go NCBI, just search Google or write NCBI and you'll get there. You see in here, you have multiple databases. If you click on this, you can go to gene. And if you look for periostin human gene and do the search, it comes out exactly the information I showed you. And then if you take one click on this guy, it will obtain you to the page where you see the gene information. Scrolling down will show you the transcript I showed you in one of the slides. If you click on the 006, you get into this page. Without clicking, just put the mouse there. Passing the mouse over this, it will give you the information. So this is gonna be the quickest way you can solve the assignment. The gene name is periostin, the title, the splice variants, the accession number, how many base pairs, the genomic DNA. So you can see it spans over 36. Oh, come on, come back. So it's, if you look, it span on 36 and 184 nucleotides. The coding is 2,511. The non-coding part, you can tell already the gene itself, how big, and then you have the coding sequence, which is 2,511, and you get 836 amino acids. If you want to click on any of these are clickable, so not allow me to do that. So if you want to click on any of these, if you click on this guy, if you want to click on the coding sequence, you can choose the coding sequence or the messenger RNA. Choosing to get the coding sequence, for example, it takes you to another page. And that's where we got before when we did the Ensembl. Okay. Cool. I think that's going to be enough for today. I will release the assignment in few minutes after the lecture, and you will have 1 week to answer the questions. It should not take you more than 30 minutes based on this lecture. Okay, guys? Stop sharing. Stop recording.